Hi, I'm Mr. Baker, and this is Unit 4, Cellular Respiration. We're talking about how cells recharge ATP for energy. Your learning goals are to learn about how living organisms obtain energy by breaking down organic molecules during the process called cellular respiration, which includes mitochondria and how they look, and the process of glycolysis, Krebs cycle, the electron transport system, and fermentation. What do we expect you to know? What enters the reactants and what exits the products of each phase, where they occur, the terms aerobic versus anaerobic, the various players in each phase, how much ATP is regenerated each phase, and the entire total for the whole process, and about a molecule called ATP synthase and something called chemiosmosis. The first section, we're going to learn these things, so let's get to it. I highly recommend that you watch this video that I'm not going to have time to show you. All living things need energy. Two pathways to releases. If you do not use oxygen, it's anaerobic. The advantage, it's fast, but it's not very efficient. You can use oxygen, that's aerobic. It's way more efficient, but not as fast. Living things probably use some combination of both. These processes occur in the mitochondria. The mitochondria has an outer membrane, an inner membrane with some folds, and fluid inside that inner membrane. Better described with a crumpled bag within a bag. The outer bag is the outer membrane. The inner bag is the inner membrane that's all crumpled up. All those folds and crinkles are called cristae to give it more surface area. And there will be some fluid in that inner bag, and that is called the matrix. You also need to know about ATP, adenosine triphosphate. We're going to call that a fully charged battery with 3 phosphates. ADP is a partially charged battery, 2 phosphates, T, tri, 3, D, di, 2. You should be able to know and recite the equation for cellular respiration, which is C6H12O6, or glucose, plus 6 oxygen. The arrow means yields, 6 carbon dioxides, 6 waters, and a release of energy. Cellular respiration requires fuel and oxygen. You're going to store energy in the chemical bonds of sugar, protein, and fats, and we're going to break those bonds to release the high-energy electrons that are captured in ATP. Like this bison that eats stuff, breathes in oxygen, releases ATP, and releases water inside the body and breathes out carbon dioxide. You're going to eat food, digest it. You're going to take those small nutrient molecules, dump them into the bloodstream, and the only one we're going to worry about is glucose that gets to the cells. Once glucose gets to the cell, now we can start to release some energy. There are 3 phases: glycolysis, Krebs cycle, and the electron transport system. You are expected to know the players: glucose, which is a 6-carbon sugar; pyruvate, which is half of that, a 3-carbon sugar; NAD and FAD, which are the electron pirates, and they always steal 2 electrons because that's what pirates do, steal things. You'll need to know that NAD plus is when it has not stolen electrons, and NAD H is when it has stolen 2 electrons. FAD H, no electrons. FAD H2 has stolen 2 electrons. You need to know ATP and ADP, which we previously discussed, and the fact that you breathe in oxygen and breathe out CO2. The overall equation: glucose comes in, we do glycolysis, get some ATP, we do Krebs cycle, get a little more ATP, release some CO2s, we dump it into the electron transport system, and we get a ton of ATP. We release water, we have to breathe in oxygen, and we can also do fermentation, but more about that later. I described glycolysis as the story of the quirky librarian. Oops. And the quirky librarian has 6 books, puts a book in on one end, then puts a book in on the other end, and then splits it right down in the middle into 2 sets of 3 books, and puts a bookend on each open end, and then takes all the bookends away. It's a very quirky thing for a librarian to do. Well, the 6 books represent glucose, and each book is a carbon atom. So there are 6 carbon atoms, and I want you to be aware of how to track the carbon atoms. When you put a bookend on, the bookend is a phosphate. You put a bookend on the other end, that's another phosphate. And we have to invest 2 ATP. It costs us to get this process going. We're going to split it in 2 sets of 3 books, and then when you put a bookend on each open end, NAD shows up, steals 2 electrons, and hooks the bookend on for free to kind of cover itself to follow our silly story. And then you're going to remove each bookend. That's 4 phosphates, so you make 4 ATP. And then you've got 2 pyruvates left at the end, which are 3 carbon atoms. Here's a handy-dandy chart telling you what's going on. Sorry, that's a table. Glycolysis in the cytoplasm. It is anaerobic, no oxygen's being used. Glucose, 2 ATP, 2 NAD plus enter. 2 pyruvates, 2 NADHs leave, and we have to consider our products of ATP. They get their own separate little column there. We make 4 total, but we invested 2, so we have 2 for our net gain. There, in normal times, would be a quiz over the Quirky Librarian story. Glycolysis, I want you to use the table I just introduced in the previous slide. It takes place where? Cytoplasm. Input, glucose, 2 ATP, 2 NAD+. Output, 2 pyruvate, 4 ATP, and 2 NADHs. There's a nice little video here, but I don't have time to show that to you. You need to look that up on your own. It gives you a more detailed look of what's going on in the molecular changes. Again, here's the table. I already ran through that once. That takes us to quiz 4.1b, which will be the official terms to glycolysis. The second phase is the Krebs cycle and occurs in the mitochondrial matrix. The simple form that we want you to know: pyruvate, you're going to peel off some carbon dioxide, you're going to make some NADs, and you're going to make some FADs. Each glucose molecule requires 2 turns of the Krebs cycle. You're going to put 1 pyruvate in, and you get 1 turn. We have 2 pyruvates left at the end of glycolysis, which means 2 turns. Ergo, each glucose, 2 turns of Krebs. There's another video embedded, which you can watch on your own time. This is what I want you to know. For each 2 turns of the Krebs cycle, this is 1 glucose going in, we yield 6 CO2, which you're going to breathe out, to ATP. So we make a couple ATPs. We make 8 NADHs and 2 FADH2s. Oh, fun fact, fun fact, fun fact. It's time for a fun fact. When someone loses weight, where does that mass go? It's exhaled. The 6 CO2 exhaled during the Krebs are the remnants of the mass of fat tissue broken down and used for energy during weight loss. So someone who's lost, say, 20 pounds, has breathed all of that out. Probably a good deal of it while they're doing exercise. Krebs cycles are always represented as a circle. So if you see a circle, you can pretty much be sure that that is the Krebs cycle. Here's a more simplistic version with what's going on, but we put that into a table for you. It's Krebs. Where does it take place? The mitochondrial matrix. Is oxygen present for our purposes? Yes, this is aerobic. What enters? 2 pyruvates, 8 NAD pluses, 2 FADHs. What exits? 6 CO2s that you exhale, 8 NADHs, 2 FADH2s, and how many ATPs do we make? 2. And that would be time for a quiz. Hope you learned something and study hard for your quiz and test. Have a good day.